So good morning, everybody. It's my pleasure to welcome Emanuela Corsini today that will present a webinar on the immunotoxicity of PFAS substances, which are very well-known substances now, unfortunately. So Emanuela has a PhD in food and environmental toxicology. She's a full professor of toxicology at the University of Milan in Italy. And her research focused on refining alternative in vitro immunotoxicity tests and elucidating molecular mechanisms of immunomodulatory compounds. Her work includes studying allergens, irritants, pesticides, endocrine disruptors, and nanoparticles. She has authored over 240 publications and contributed to national and international projects. She's active and very active in scientific organizations like ESAC, EFSA, EMA, the cosmetic SSS, CCS, OECD, and she serves also on editorial boards of toxicology journals. She's the Secretary General of EUROTOX, which is an international toxicology society, since 2019, and she now the president-elect from EUROTOX, and she has been elected in 2023. So thank you very much, Emmanuel, for accepting to give this course. So for the participants, please put your question in the chat. And then for the last 15 minutes, I will read the questions to the chat. If we have still enough time and no question in the chat, you can ask your question directly if you want. But the question will be read in the chat. But first, please use the chat. And please shut down your microphone. Okay. Thank you very much. The floor is yours, Emanuela.
So thank you very much and welcome everybody. As you see, the title is Immunotoxicity of Perfluorinated Compounds. And first of all, I have to thank, of course, Mark, but also the Education Subcommittee at EUROTOX, because they are putting together these interesting webinars. So it's not just mine, have been other in the past and there will be other in the future, which I think is an important activity for EUROTOX. They show that besides having a very successful Congress, we also do something else, and thanks to the Education Subcommittee. So many things will come, so please also stay tuned. Now, first of all, as usually, the second slide is a conflict of interest statement. To be honest, I don't have any conflict of interest to declare, with the exception, with the bias of immunotoxicity and how is important the adverse effect of xenobiotic on the immune system. But besides that, honestly, I do not have any conflict of interest. Now, as a layout of my presentation, I would like to start with a brief introduction on the uses, the environmental contamination, the human exposure to this huge, we will see, class of compounds, then briefly touch on the toxicity, the adverse effect that has been associated with the exposure to these chemicals, while I will focus the majority of my presentation on the immunotoxicity of perfluorinated compounds, some in vivo epidemiological evidence, and in vitro immunotoxicity, because as Mark pointed out, the laboratory we are quite interested in vitro immunotoxicology and the different use under the umbrella of in vitro immunotox. And of course, some summary and conclusion. So let's start with the introduction to PFAS. Now, if you look at the definition, the per and polyfluoroalkyl substances are chemicals that do contain multiple fluorine atoms attached to an alkylic chain. Very broad definition. So if we stay with this definition, you may find that we have more than 10,000 chemicals that actually fit under this definition. And immediately it is clear how it is impossible, or honestly, if we do not find alternative way to define the risk that this class of chemicals may pose to humans and to the environment. So it's a huge class. They include some compounds that are present in our daily life. So if you think Teflon, if you think Gore-Tex, to name some, but then perfluorinated are also, we can find some pesticides, we can find some pharmaceuticals pharmaceutical. So it's a very huge, so probably there is the need to redefine and to follow sulfonate and the perfluorooctane acid. Now, these are the ones that immediately we know that belong to this class, and these are the chemicals for which we honestly have the majority of data. Now, so there are different ways beside the presence of fluorine and carbon. Now, depending within the perfluoroalkyl substances, this can be divided into big group. The one that we call the long chain, meaning compounds that contain more than 6 carbon atoms, and the short chain. These are in this table you find a list of some of these compounds, which of course include PFOA and PFAS. Now, what are the characteristics of these chemicals? If you look at the structure, they have a chain that provide an hydrophobic portion to the molecules, while we have an hydrophilic portion. So due to this characteristic, these compounds have a very good water and oil repellent properties, and they have been used and they are used as a surfactants, as dispersants. The use of this class of compounds go back to the 1940s when they were first introduced. The other characteristic of these compounds is linked to the strength of the bond between carbon and fluoride, which is one of the most strong bond that we can have, which have immediately an implication in terms of environmental contamination and persistence. So these compounds are not easy to degrade. They are not really undergo any metabolic process, which give to these compounds also the name of forever chemicals due to their persistence in the environment. It has been estimated, of course, that some of the perfluorinated compounds may take over than 1,000 years to degrade. And this poses the problem for the environmental, but also for humans, because these compounds can move within our environment from the place where they are synthesized, the compounds they contain perfluorinated compounds, and then we can find they can contaminate the water, the soil, and so they come back to the food and drinking water and then arriving to humans. So these compounds are characterized by mobility, persistence, and bioaccumulation. And this is actually what raises a lot of concern for this category of chemicals. Now, we have to admit that as it was for asbestos in the Industrial Revolution, these compounds really have unique properties that we also, as consumers, like quite a lot. These compounds can be found, if you look in these slides, in many daily life products. But then, of course, when we think on how we can be exposed to perfluorinated compounds, of course, if in the working setting you work in occupations where PFAS are used, including firefighting, chemical manufacturing and processing, we have exposed workers. Drinking water contaminated with perfluorinated compounds, eating foods contaminated with PFAS, including fish. Especially for children, swallowing contaminated soil or dust is also a way of exposure. Breathing air, because either we found in the particles or some are volatile, we may also have an inhalation exposure. Even if it's not completely sure what is the impact of the presence of food in that are present in the package and the leakage from either to our hands or to the food. This strong is the contribution is still under discussion, but this could also represent an important source of exposure. So many ways that we can be exposed to perfluorinated compounds, and actually we are all daily exposed to this class of compounds. If we look at the human level of exposure, then, and there are of course many articles that you can find that they have measured in different settings. We can say very broad that for the general population, the plasma or serum level of PFAS are in the range of nanogram per milliliter of serum of blood. Well, if we go in a very highly contaminated area or in some industrial setting, levels in the microgram per milliliter range has been also described in the literature. And these are the value from an EFSA document where the median blood concentration in the European Union, if we look in the adults for PFOS, is 7.7 nanogram per milliliters and PFOA 1.9 nanogram per milliliters. So this is more or less the level. The other interesting thing, now I told you, so it's a huge class of compounds. Then, of course, I mean, it's not that all are found in our body, but the first, if you remember the slides where I have the long and the short chain, the first 4 long chain account for more than 50% of the blood concentration of the PFAS, and they also account for almost 90% of the residues that can be found in food. So meaning that the long chain are contributing quite high to our exposure, but these are the level. Now, of course, the concern is not because, I mean, they are persistent in the environment, but the concern is for the adverse effect. Now, in these slides, you can see many different targets in female and in male. The one that they have a straight line are the one for which there is the strength of association is more strong, meaning that we have more epi study, but also supported by animal study that show an association between PFAS exposure and the adverse effect. And you see here thyroid disease, cholesterol level, cardiovascular disease, liver damage, kidney cancer, but I will also come back. The one with the dotted line are the one for which there are some studies that show an association, but the strength is not so strong. If we want to see in a different way the same concept, we see that we have evidence quite strong of association between reproductive effect, which include decreased fertility, increased high blood pressure in pregnant women. There are effects on to the developmental effect and delays in children, including low birth weight, accelerated puberty, bone variation, behavioral changes. Increasing cholesterol, there is also some studies that show an increased risk of obesity. Interference is unfortunately, they are also endocrine active compounds, and so they can interfere with the body's natural hormones. But then if we look at the last 2 effects, there is a strong evidence on the ability of PFAS to decrease the resistance to infection, including a reduction in response to vaccination. The other link that has been done is with some cancer and for some perfluorinated compound, there is an increased risk of some cancers, including kidney, prostate, and testicular cancer. Now, if we think of these 2 last adverse health effects, they can be directly or indirectly linked to our immune system. And these, of course, bring me to the second part of my presentation, where on PFAS and immunotoxicity. of course, the question is why. Now, if we look at overall the data, even if we have to admit that not all regulatory agencies use use the immune system as the critical endpoints, but we have to acknowledge that the effect on the immune system were observed in several studies, often at lower levels compared to other adverse effects, including liver toxicity, change in thyroid hormones, which made the immune system a critical target of the adverse effect of perfluorinated compounds. The effect of, especially the main association with the strong evidence is PFAS, immunosuppression, reduced resistance to vaccination. There are several studies where they address the response to vaccination, and in this study, at least one measure of the anti-vaccine antibody was shown across multiple studies with evidence in different age, from developmental, childhood, and adult exposure have been reported. As I said, there are also studies show the association with an increased risk of infection. And as I also mentioned, and this is very recently, and I know that Mark was involved at the hierarchical level, PFOA has been classified as group 1, meaning carcinogenic to humans, while PFOS as possibly carcinogen, which is in group 2B. There are also some studies that show association with allergy and autoimmunity. So many direct and indirect effect link to our immune system. And so as I said, not all agencies agree, or better to say, choose the immune system. In Europe, the European Food Safety Agency actually identified as a decreased response to vaccination in children as a critical endpoint. And after benchmark modeling of serum level of the sum of the 4 PFAS, the 4 long chain that I mentioned before, they have estimated the corresponding daily intakes and establish a tolerable weekly intake of 4.4 nanogram per kilogram of body weight. It is clear that with these levels, also any other adverse effect, being this one the most sensitive, which means occurring at a lower dose, is going to protect also other adverse effects. Now, the immune system as a critical endpoint. Now, in vitro immunotoxicity, because so this is the main what in my daily laboratory work. So I will be presenting, then I will also refer of some way where you can find more details, but I will be commenting and presenting results from 2 projects. One that ended, that was actually sponsored by EFSA, and it was on a case study on the new approach methodologies focusing on PFAS immunotoxicity, while the other studies we have been granted by the Italian government with the focus and to understand the possible difference between short and long chain PFAS. So in total, I'll be showing some data reflecting 7 perfluorinated compounds. We have the 4 long chain that I mentioned several times, and then we have 3 short chain. One of the striking difference between the short and the long is the different the half-life. One of so this is also why we have such a big concern, is because if we look at the half-life of the long chain PFAS, we are talking of more than 3 years of half-life. So these compounds, really, we are daily exposed, but then we tend to accumulate over the years we increase the exposure to this class of compounds. So these are very, it's what is of concern. If we compare to the short chain, they have days or at least, or more or less 1 month. So really they have a different behavior in terms of internal and half-life for these compounds. Now, the way we decide for the concentration to use for the in vitro study. So we of course we use a non-cytotoxic concentration, and we use a range that go from 1 nanogram to 10 microgram, where the nanogram range is relevant for the general population. The microgram level is more relevant for occupational setting or people living in a highly contaminated sites. So these are the concentration. Now let's start, let's say, what the focus within the EFSA project. The EFSA project want to have more information to fill some of the data gaps. First of all, to get information on the mode of action of the 4 main perfluorinated compounds, with a focus on the antibody response, because the decreased response in vaccination was a critical endpoint. Now, the approach that, and if you are interested, then I will give you the reference for the EFSA report, where all the SOP that we develop and use within the project are actually available. And there is also more details on the results that we obtain. The goal was the antibody response. When you think of the antibody response, we can divide the antigen, and the response could be T-cell independent antibody production, while we know that for the majority of the antigen, we have a T-cell dependent antibody production. Of course, when we think of the T-cell independent, so everything's focused on the B cells. When we think of the T-cell dependent antibody response, then we have more actor. We have the dendritic cells that are required for the antigen presentation to the T helper cells, which then crosstalk with the B cells, activate the B cells with antibody production. So the way we propose to approach to the antibody production, beside the assessment for leukotoxicity, was to dissect the antibody production, meaning that we study in isolation, let's put in this way, the maturation of dendritic cells, the effect on T helper cells, proliferation and differentiation, the activation of B cells. We also look for the sort of polyclonal B cell activation, and we measure the immunoglobulin release, and we also have methods for in vitro primary antibody response, which still require some optimization. To answer the question on the mode of action, we use RNA-seq, and the project has also an important in silico component that I will briefly mention, because I'm not an expert in computational methods, but we use PBK models for fate and distribution to extrapolate the in vitro to in vivo, to the in vitro concentration to in vivo dose, and then we use the universal immune system simulator to simulate based of concentration of plasma concentration of PFAS to simulate the decrease of the response to vaccination. So it was a very nice project, a different group, but we generated really a lot of data. First of all, of course, I mean, this cannot be done only in my lab. So this was a collaboration with the University of Wageningen, with the University of Catania for the Universal Immune System Simulator, and with ESCI Lab for the in silico and the in silico part for the Chi Vive and so on. Now, as I mentioned, so this is the reference where you can find additional information and the SOP. To make a very strong, I mean, a very, to make sort of the long story short, So these are the main findings that we have. First of all, PFAS, but this we were expecting, can directly affect immune cells functionality. And we found effect at a different level. We found effect on the dendritic cells maturation and with a decrease in MHC class 2 HLA-DR expression, a decrease in some cytokine release, such as interleukin 6 and interleukin 8. We found an effect on the maturation and differentiation of T helper cells. We couldn't really, we see a decrease mainly in T helper 1, but not only, because also the Treg were decreased as well. So there was a decrease in T cell proliferation, a decrease in T helper 1, a decrease in some cytokines, interferon gamma, interleukin 4, interleukin 17, a decrease in the Treg. So they can broadly affect the T cells. But we can also find effect on the B cells, where we have a reduction in the release of anti-KLH IgM antibody and a reduced release of IgG and IgM, of a release of immunoglobulin. So really, with a clear indication of immunosuppression. Some of the effects were observed in the nanogram per milliliter concentration, which meaning that these are also relevant for the general population for our exposure. And of course, I forgot to mention all the study and the data have been generated using either human cells or primary PBNC, peripheral blood mononuclear cells, obtaining from healthy donor male and females. Okay. Because I forgot to mention, just to show you some data, because so these are more new data. So this is the protocol for the maturation of dendritic cells. So we use the THP1. THP1 is a promyelocytic cell line that being promyelocytic, can be differentiating in many cells, so the myeloid lineage. We use a cocktail of a protocol that has been established in the literature, where they use a cocktail of interleukin 4 and GM-CSF for 5 days. Then to get immature dendritic cells. Then we expose the immature dendritic cells for 24 hours to increasing concentration of the perfluorinated compounds. And then we look at the effect on the maturation of the dendritic cells, where you add a cytokine cocktail. And we look at the surface marker expression by FACS analysis after 24 and 72 hours. And then we also measure cytokines that I mentioned before. Now, so these are the results, and this is what I want to make a comment. Now, I've been only presenting data on MHC class 2 after 24 and 72 hours. So the one is the maturation that we achieve in the vehicle-exposed cells. Increasing concentration, and especially for this specific marker, the upregulation, the optimal is the 72 hours, not the 24. So if you focus on the bottom, you see that the different long chain, especially PFAS and PFOA, have a clear dose-response decrease in the expression of HLA-DR. But if we compare with the short chain PFAS, basically the short chain has no effect. Okay. So this was the first new results that we have. Then we look for the T-cell independent antibody production. Here we use a protocol that was published in the literature, and so these are the name and the year of the publication, where they are actually studying, they set a system to measure the specific B-cell, in a sense, polyclonal of a B-cell receptor independent B-cell activation, where you use another antagonist of toll-like receptor 9 in the presence of interleukin-2 to promote immunoglobulin release. So also here is a longer protocol compared to the dendritic cell maturation. So the same protocol, 24 hours pre-incubation, and then we stimulate the cells for 6 days, and after 6 days we measure the total immunoglobulin IgM and IgG release. These are the results. This response was obtained using PBNC. So where we can see also answer the question, is there a gender difference in the sensitivity to the adverse effect of in vitro of perfluorinated compounds? So on top we have the male and on the bottom the female. Also here you can see that more or less with the, so the pot there is, if you want, so they are not, I mean, the effect so is not equal also among the four long chain PFAS, especially like if you look in males. In the female, more or less we can reach the same, they have a similar potency, the four long chain, but it seems to decrease, so to be more sensitive, the female compared to the male. But the message that I want to give here is not, so we could reproduce the decrease in the antibody response, but also if we compare long versus short, also for these parameters, which is more apical and more, they're all relevant for the immune response, but the immunoglobulin production, of course, is more apical than not the surface marker expression, but also here the short chain are less effective. So here we are talking of nominal concentration, because so the measure is what actually we add in the cultural medium. So we took advantage of an in vitro mass balance model, the Armitage model, actually to have a little better, it's an in silico, so we didn't really physically measure with an analytical methods, the presence of the perfluorinated in the different compartment. But so we use these methods to predict what could be the distribution, meaning the cell fraction, the amount free in the culture medium, the amount bound to protein, and so on. And this come to me what is, I think, an important observation. Now, in this table, I have reported the 7 perfluorinated compound, the long and the short, the Cas number, the molecular weight, the log P, and the mass distribution. Now, there are, if you look, the percentage of PFAS, or the different PFAS that actually go into the cells, there is a strike difference, okay? So we have a maximum for PFOS, and we have the minimum for the two PFBS and PFBA, where only 0.6% actually get into the cells, okay? And it is also interesting, if you look at the water, so while the long chain are basically the major are either within the cells or bound to albumin, the short chain, they have some are still bound to albumin, but some are more present in the water fraction. So the distribution within the different cellular, let's say tissue cultural compartment is quite different. So we ask, but is there a correlation between this mass fraction distribution and the decrease in the antibody production once following like an exposure in our in vitro system to 10 microgram per milliliter, which is the highest concentration that we tested. And indeed, and so this is the results. Either we use a parametric and non-parametric correlation. If we look at the Spearman, the one that show like the best correlation between the two. We have a high, but in both cases, it's significant, the correlation between the decrease, so more intracellular PFAS we have, the higher is a decrease in the antibody response. Okay, so and this provide a very simple explanation on the lack of effectiveness of the short chain PFAS, or at least for the last two, they show basically no effect at all on immunoglobulin release. Mode of action, also here very short, because I mean, can you guess the amount of information that you have out of RNA-seq? But what we know from the literature first, there are, I mean, if you look, so there are indications of the ability of this class of compounds to affect many signal transduction in the pathway, to interfere with many hormones. Of course, due to the structure that look like a fatty acid, of course, they can be agonists of PPAR alpha. Then there are indications of the involvement of PPAR gamma, CAR, estrogen receptor, androgen receptor, glucocorticoid receptor, pregnan X receptor, NRF2, NFKB, interference with the thyroid hormones, homeostasis. So really, it's so they can do many things to our cells. So we look at the RNA-seq for the effect on where we are. So we have used PFOA and PFOS on the maturation of dendritic cells, on T cells, and on B cells. And what we are currently doing is then expanding. So we have selected some genes, and then to see if also the other two long chain PFAS could have a similar modulation. So what are many genes were up or down regulated. When you go to the most pronounced signaling pathway, and also if PFOA or PFOS which one was more affected, are reported in these slides. Now, if we look at the signaling pathway, we have two main signaling pathways pop up as the one modulated. We have the PPAR alpha and the glucocorticoid receptor. Okay. For some, PFOA was the effect was more profound compared to PFOS, while, for example, in PBMC and PPAR alpha was more affected by PFOS. And then there are some gender, male and female. So, but overall, we have like two main pathways, the PPAR alpha and the glucocorticoid receptor. Now, since I have a bias here, okay, not only in immunotox, but also on we all have somehow like a signal transduction that we may like more. I have two PKC and the glucocorticoid receptor. So we performed experiments in which we block the glucocorticoid receptor using mifepristone, and these are the results. So once again, so we have the 4 long chain that to a different extent, so this is 10 microgram per milliliter, which is the highest, more effective concentration that we have. And you see they all, both in male and in female, decrease the release of immunoglobulin. In the presence of mifepristone, the inhibitory effect was completely lost. And actually, we have even an increase in the immunoglobulin production, which indicate that there is also within the system a regulatory role for the glucocorticoid receptor in modulating the cell activation and the immunoglobulin production, also in resting in normal condition. So this study actually, of course, this need to be confirmed. There are, and also, of course, sorry, I forgot to mention, dexamethasone does exactly the same, and where we use dexamethasone or rapamycin as a positive control for the in vitro study. So the message here is that one of the signal is not only one, but one of the signal that is actually involved have been activated by the perfluorinated compounds. If we modulate the signal transduction, then we can modulate the response, which indicate that indeed the glucocorticoid receptor is important in the immunosuppression that we found in vitro. Now, so I get in a close. Okay, so then I think I'm in perfect time. Summary and conclusions. Now, I will have two type of conclusion. One specifically related to immunotox. First of all, in vitro human immune system models can be, which is sort of easy to assess in primary cells, offer the opportunity to study PFAS immunotoxicity. You can buy a Buffy coat from hospital, and this is what we did. So I hope that I know that I haven't shown you too many data, but really if you go to the report and to some to the upcoming publication, PFAS can directly affect immune cells functionality. We can confirm that the PFAS in vitro can inhibit antibody production, which actually, to us, is a demonstration of the relevance of in vitro models. As I said, there is a room for improvement of in vitro primary antibody response, but actually I haven't shown you the slides. For some parameters, females appear to more sensitive, even if overall we didn't really see a strike difference between male and females. When it comes to potency, if we put together all the data that we have, beside the one that I show, PFOA and PFOS seems to be slightly more potent compared to PFNA and PFHXS, and definitely the long chain are more potent than the short chain PFAS. By RNA-seq, we demonstrate that there is a role of PPAR alpha in dendritic cell maturation. There is a role of PPAR alpha and the glucocorticoid receptor in T helper cells activation. And with the use of in silico models, including in vitro distribution, PBK, Kivive, universal immune system simulator, we really, depending of course then the models, the parameter that you select, but we were able actually to come very, very close to the EFSA, the weekly or daily intake, also using the decrease in the immunoglobulin production. We come very close to what has been calculated in vivo, depending, as I said, on the parameters, like if you take into account or not to the protein binding, then we may be more conservative, meaning that we arrive to a benchmark dose lower to a tolerable daily intake, lower compared to the one estimated by EFSA following human in vivo exposure. But it means that the system really provides a reliable and relevant information for humans. Now, the other conclusion that I want to make is that because are we at risk or not? Now, if we think, what EFSA wrote in their opinion and taking into account the plasma concentration, actually, there is some concern because a considerable portion of the population is actually the exposure, the daily exposure exceed the tolerable weekly intake, which means that they may be or maybe not, but this is not, of course, an optimal situation. The other big problem of this class of compounds is the paucity of data that we have. Not only in vitro, you may find more not consistent to all possible endpoints, but you may find data for approximately 100 PFAS, but when we go like from in vivo animal data, we have some also there, not equally, but for 15 perfluorinated compounds. If we think that we have more than 10,000, so there is a long way. So there is really a different strategy must be put in place. And of course, so my conclusion, without this kind of information, making an informed risk assessment decision is still, is difficult. And not to mention that worldwide, not everybody agree on how to conduct a risk assessment. With that, of course, I thank once again the different groups that has been involved in this project that I mentioned before. And of course, I thank you for your attention, and I will be glad if you have to answer any question. So once again, thank you. Mark, I cannot hear you. Open the microphone.
Exactly. So thank you very much, Emanuela. So I've got a question in the chat. I will start with Maria Davoren, that she had two questions. First, she's interested to know what are the factors that contribute to the higher serum PFAS concentration in men compared to women.
Okay, so this could be depending, on the food intake, so that maybe male maybe eat under some circumstances more, depending on actually also on the work that they do, so they may have a higher worker, occupational exposure to perfluorinated compound. This could be a possible explanation, while some studies show higher level in the male. Now, what I don't know if the kidney uptake, because the problem of these compounds that they are released in the urine, so they are, glomerular filtration, but then there is an active, there is a reuptake at the tubular, proximal tubular levels. If this is more efficient between male and female, that's, to be honest, I don't know.
Okay. Thank you. I got a second question, a little bit longer. So Maria, she's asking, so the mass balance data demonstrates that the actual PFAS exposure to the cells in culture is significantly reduced due to confounding factors like binding to albumin, so that the actual concentration may be far lower than nominal. So confirmation of actual exposure concentration is an important consideration. How easy is this to incorporate into test methods?
Okay, so this is not so difficult because you have, as I mentioned, so when you want to do like the extrapolation with in vitro to in vivo, you can take into account the albumin, which is mainly like the protein on albumin level. Okay, so this can be considering. Now, if you take this into account, as you correctly say, we have, so the free for it is, in a sense, is lower, but then these compounds can be highly uptake by the cells. So if you adjust for the albumin binding, then you better can rank the potency of your compounds, and this can be taken. The in vitro system, the key vive models can take this into consideration, and if you take this into consideration, then we arrive to estimate a daily intake which is absolutely in the same range of the one established by EFSA. If you do not account for the protein binding of the four long chain, then we estimate a lower daily intake. So meaning that we are more protective. So if you take into account the protein binding, then you can actually be more closer to the value that has been established by EFSA.
Okay, then I've got a question from Sofiane. Sofiane, she was curious to know if you were using the liquid direct in a testing system, or if you're using a gas chamber for exposure to PFAS.
No, they're all okay. So they are soluble in DMSO. They are not really the one that we tested. They are not volatile. So, and we use a traditional subculture system because using, like, if you think of exposure of the immune cells or the PBNC, so then is a blood, whatever is present in our blood. So no, we put directly in the cultural medium. And then when we found, when we do the mass fraction, there is basically no, these compounds, they don't have, they are not volatile, the one that we tested. There are few that are considering PFAS that are volatiles, and so these, of course, need to be careful when you test these compounds. You need to know upfront, because otherwise then you may have exposure of the researcher, and also you need to have a different system of cellular exposure in order to avoid dispersion of the volatile. But these are not volatile compounds.
Thank you. One question for Marie-Charlotte Bernier. So based on these studies and considering the family of PFAS is huge, what is your personal opinion on the toxicity of not common PFAS, meaning not the 13 most known?
Yeah, okay. So this is a $1,000 question, because it's really, so there are so many, we need to find a different way trial to predict. Of course, if we can demonstrate, so they have a very, if the mode of action is actually overlapping for the different PFAS, or how to prioritize the chemicals, because also this is the question. And not to forget that the immune system is very sensitive, but not necessary. So we don't know if there will be like some PFAS for which, the immune system is not more sensitive compared to the liver or to the reproductive system. So this need to be somehow demonstrate, because these compounds have different targets, not just the immune system. How can we screen? So we probably, because, if the glucocorticoid receptor is important, or the affinity for the PPAR alpha, can we use in silico to predict a sort of endocrine disruptome? So could be these screening tools, meaning you know the structure, and then you look for the possible binding for the different receptors. So this may be a way to prioritize the chemicals, but of course we need to be sure that we are not missing other effect, because the hormonal system the PPAR alpha is definitely pop up in many different studies, but then if you go high, then you have a more oxidative stress, NRF2, NFKB activation, so that not necessarily are covered by investigating the endocrine disruptome. So I don't really have a solution. What I may suggest, if you want to investigate in vitro the immunotoxicity, then we have more than one assays that actually identify the immunotoxic effect. More easy. No, it's not really, I would not say more easy than the other. Like if you use the THP1, which is a cell line, maybe it would be like in a sense more easy, because you can buy the cell lines and then use. If you use PBNC, then you can frozen, but then you need, so there is high variability. But of course, this also is important to understand the response in humans. So there are, all the methods that actually we propose, they seems to identify the immunotoxic potential if you want to screen there. But like once again, 10,000 chemicals. Also, phasing out animals, the time will be impossible. It's really a huge time. So a different strategy. In silico, we should try to see if we can somehow use in silico methods to predict something. Otherwise, then we will not be able to deal with 10,000 chemicals. I don't know if someone has other suggestions, because it's still it's under discussion how to prioritize the chemicals.
I think the question that's quite similar from Émilie Grosdidier is whether a study of the possible cumulative effects of the different PFAS to which people are commonly exposed. To test, the test show one molecule is able to trigger effects. What about 2 or 3 or more molecules at the same time?
Yeah, I mean, the problem of mixture, this is absolutely true. We are not exposed just to one. We think, like if we think of the four in a long chain, that is a sort of a sum, 1+1. Okay, it's more like a sort of possible additive effect is what we may expect. Okay, so what would be like interesting? Of course, then any mixture, I mean, is representative of something happening in a certain point. Or, I mean, the other possibility is to use like a very low concentration and then add the six, or there are study and actually one done which has been done like in Germany where they take contaminated sites where they have also oil and they have done some studies where they demonstrate that there is indeed an additive effect. So where they use so they know like the contaminated sites, the level of different the perfluorinated that they measure, and so then they reconstitute in vitro the same mixture to see the effect. And so the suggestion is a possible additive effect, which the sum could then could be of an explanation. So meaning that is not really like a synergic effect, is more an additive effect. Of course, like if I have to look at the 7 that we use, we have 3 that basically they don't do anything, while I left 4, but we have not in our experimental system. So this would be interesting to do, and to mix the perfluorinated and see if we have really an additive effect.
What is not we don't have as a tool, like for the dioxin compounds, related compound, we have a potency assay. And here you don't have a potency assay to see what could be the potency of each of the molecules.
The only way you can do like a potency is just like, I don't know, to refer either to PFA or to PFAS, and so this will be like the most potent, and then you can rank the other based on the most sensitive parameters, or you can do for the different endpoint that we have. And then, because, so this is something that, I mean, when I say like PFOA and PFOS, and overall, if I take all the effect, they have similar potency, while the other two are slightly less potent. So where for potency, you're considering the lowest, the effect that you find at the lowest concentration.
So I've got some more questions. So one from Yasmin Farid. I would like to know your opinion on PFAS in respect to inflammation.
Okay, so the one of inflammation. So this is also like a good point, because there are like some study that show that you may have like a sort of possible, no, but then the correlation with autoimmunity and allergy is not necessarily linked to inflammation. The possibility to see inflammation is when you go high in concentration of doses, like if you go in vivo, and then in this situation you may have an increase, also like an indication of NFKB activation, which of course a potentiation of the response. For some, this, but then you need to go in a little higher concentration, not really in the nanogram range. Then you may see an increase in NFKB and a possible some inflammation coming from there. So this is to my knowledge, because I mean in the system that we use. Where you have like immune cells, we do not really see an increase in pro-inflammatory cytokine production, because I mean what we see is more a link to a decrease, to an immunosuppressive effect. So we do not really have indication of an inflammatory effect in our experimental. Looking in the literature, where they do see some is like oxidative stress and then inflammation, but higher doses or concentration.
I've got three more questions. The question from Yasmin. No, the next question is from Ivoni. The US EPA recently identified carcinogenicity and not immunotoxicity as the critical endpoint for PFAS. What is your view on this?
Okay, so I mean, it's like depending what you look for, like in animals versus humans. So because the two definitely, like if you think of liver cancer and in experimental animals, it's really something that will pop up. So this is absolutely true. We should not forget that the immunosuppression is one of the key characteristics of carcinogens. So it's not, then of course, is one of the mechanism by which a tumor can grow. But the immune system, if you are exposed to an immunosuppressive compounds, this is increase the risk of cancer. So this is what I think of the link between the immune system, immunosuppression, and increased risk of cancer. Then, of course, I mean, depending on the study, the immunological endpoints that they have considered, not like the status of the immune system or the functionality that you may include in your study could provide a different answer. So it's really this need to be put. I mean, I don't know exactly which il data they have, or like immunological data they have considering. But to me, I mean, an immunosuppressive compounds increase risk of cancer. So this has been also shown by the NTP studies, where even if there is no any osteoresistance assay that can be linked to a specific immunological status or functions, then there is a correlation between immunosuppression and cancer. There is no correlation between immunotoxicity and genotoxicity and mutagenicity. So it's really as more like a recognition of transformed cells and elimination. Of course, then we can enter the tumor immunology, whereas the immune system could be a double sword, depending on the context, especially like the innate immunity actually could favor cancer growth.
So one question from Shrouk Sherif, if PFAS has more damage to immunity even in lower doses, would that affect the vaccine production in future?
Yeah, so this could be like a good question, because probably if we
The vaccine
Yeah,
response.
no, I'm just like like the dose of vaccine should be somehow adjusted based on. So, but then like do like a personal vaccination is not really something feasible to do. But we should also considering that in the future we are expecting, since these chemicals are going to be somehow regulated. So the exposure in the future should be low.
Yeah,
Okay.
it is
So
already
this
low.
should not really be in the future of high concern.
Yeah, because plus the vaccine is a very complex situation, because it's a very immune personal response. I mean, some people
Yeah.
don't have the good T cell repertoire. For example,
Yeah. So
the flu vaccine is not working very well in France this year. We don't know why. So you see, it's
yeah.
very complex.
Very complex, yeah. So then I found like a difficulty to think personal. I mean, everything's. I mean, we can think of everything, of course, like for the future, like a sort of based on your immune system and immune at this age, because then it's going to change when you get older. So the best regimen, the best adjuvant for you to have an optimal response and protection. So which is of course important.
So I have two more questions from Sadia Kerdin Romer. So I'd like to know your opinion on PFAS in terms of skin toxicity, sensitization, irritation. That's the first question. And the second question, many studies focus on a single PFAS or PFOS. What do you think of mixture? That has been already addressed. So
Yeah, address. So the
you can
mixture,
answer the first
yeah.
question.
Yeah, the first question in term, okay, so there are some studies that show an association with an increased risk of allergy and also some autoimmunity. The association is not as strong as the one for the decreased response to vaccination and um, to an increased risk of infection. So there is the possibility of screwed up. I mean, in our experimental system, to be honest, because we see more like an effect on the T helper 1 compared to T helper 2, which means that, but we also see a decrease on interleukin 4. So we do not really, this need to be addressed. So the model that currently we have we can think to add an additional, like an allergens, chemical allergens, to see how the response is modulated. So, but to be honest, we have not done, so I don't really have, I mean, the only answer that I can give is that the study that show this association, but like in terms of more inside in vitro, this need to be, could be easily tested, but I don't have the data, so I don't know.
And last question, again for Marie-Charlotte Bernier. Are you planning to use your methods for other PFAS and start creating this precious data?
Okay, so I mean, so far we have from 4 we move we added the short chain, and so yes, the idea is yes to see to test some more the system. Especially for the data that I have, so we have SOP is, I would say that is, I mean, we have been using for several years, so it's sort of optimize. What we still need to work is for the TDAR, because we need to, and we are currently working to optimize the TDAR, the in vitro TDAR response, which will be, of course, quite important if we want a link also. I mean, the TDAR, I mean, is also somehow like you can link to the antibody, to the vaccine response with actually a surrogate in humans for the TDAR. So this is the way Immunotox use the response to vaccination. So the answer is yes. So we are actively working on more PFAS.
So thank you, Emanuela. I've got no more questions, and we are already 10 past 2. So it was a very successful webinar. I've got a lot of comments with a lot of thanks and a lot of congratulations of your presentation. So thank you again. It was very nice, and I wish a very nice day to all the participants, and of course to you, Emanuela. Okay.
Thank you very much, and wish you to see you all at EUROTOX, of course, and to the next webinar. So
Okay.
thank you very much.
Thank you.
Bye,
Bye-bye.
everybody.
Bye.